This lecture is part one of two for the first chapter of the book, Web of Life. And this chapter deals with a few key concepts. One is events in the natural world are interconnected, and ecology is the scientific study of interactions between organisms and their environment. And ecologists evaluate competing hypotheses about natural systems with observations and experiments and models. So it's kind of laying the groundwork for what we'll be doing through the rest of the quarter about how we ask and answer questions in ecology and how ecology works at multiple scales and also deals with the interactions between organisms with each other, within themselves, and within the abiotic environment. And this chapter uses a pretty cool case study looking at amphibians and looking at how the understanding of this really crazy deformity in amphibians has changed. How we've, through a series of experiments, been able to understand more and more about why we see this in the context that amphibian populations are declining worldwide. And that decline has a lot to do with the way that amphibians are really susceptible to pollution and other impacts. And this is a global distribution of the number of extinct, missing, or endangered, or critically endangered amphibians and the ones that are threatened. And it probably mirrors pretty closely also the distribution of amphibians. So if you look at Central America and South America, they have by far and away the most number of extinct or critically endangered species, as well as threatened and vulnerable amphibian species. Which probably has a lot to do with the distribution of amphibians as well, but nonetheless pretty interesting. And so amphibians are good biological indicators of environmental problems. And what a biological indicator is an organism or suite of organisms that their condition reflects the environment. Other examples of that include there's a number of stream indices based on invertebrates, so that the type of insects that you see in streams is a good indication of the overall health of that stream. As well as in the marine environment, there's a few indices that we use around infaunal, meaning organisms that live within the sediment, infaunal diversity indices. And so one example of that is some work done by the state that goes around and they measure the diversity of organisms you find when you drop a core off the side of a boat. It goes down, takes a big chunk of mud, and then you sieve that and count all the little critters that you find in the mud. Based on that index, Bellingham Bay has one of the lowest values of that index, meaning that our infaunal diversity in Bellingham Bay is quite low, which is a measure of the overall health of Bellingham Bay. And so generally Bellingham Bay has a fair number of issues, both mercury, low oxygen, etc. And that's reflected in that biological indicator. So similarly, amphibians are good biological indicators because their skin is permeable. Pollutant molecules can pass through them easily. Their eggs have no protective shell. They spend part of their life on land and part in water, so they're exposed to pollutants in both environments. And so it's a good example of a organism that needs a intact ecosystem to survive. And so because of that, they're great biological indicators. So even species that do not interact directly can be connected by shared environmental features, meaning you can have organisms living in the same general space but not interacting. They're still impacted by the same changes to the abiotic environment. And so as ecologists, we ask questions about the natural world in order to understand these connections. Move this here. So here's a couple of images of these frogs in question. And these deformed frogs in Minnesota have been kind of a mystery for a long time, and we'll walk through how we've increased our understanding of these deformities and the things that have been happening since then. Really being observed in the mid '90s, and still a lot of ongoing research into why this is happening. And so this chapter walks through the broad idea of ecology and the scientific process using an investigation of this particular amphibian as an example. And linked here is a Minnesota NPR study or story about the researcher that's been working on this for a long time and has written a book about it. So to understand this issue, we need to understand the life cycle of Riberia, which is the parasitic flatworm that is partially responsible for these deformities. This is a pretty common, or these parasitic flatworms with this life cycle are pretty common. I've done work with another system, actually coastal Southern California, where the Metasarcariae, the cysts show up in clams. There's a little clam in Southern California called the bean clam. It's about a centimeter long. It can just cover the beach. And in this little tiny clam that's, a bean clam, because it's about the size of a bean, a centimeter long, we found over 300 Metasarcariae in one little clam. I mean, when you cut it open and look, it's just a sea of these little tiny cysts. It's pretty gross. That was, I forget the genus and species of that. But I had an undergrad work on that when I was in grad school, and she wrote a nice little paper about it. So in this case, Riberia has a very complex life cycle. And so if we start with the bird that's the definitive host, the bird consumes prey that have the Metasarcariae in it, the cysts. Those cysts then hatch in the gut of the bird as adult parasites, where they undergo sexual reproduction and release eggs. These eggs then hatch in the environment, where they're Myricidae. The Myricidae then get taken up by a first intermediate host. Typically, I think it's consumed as well. I think they consume the material that has the Myricidae in it. Within the snail, they then hatch to Cercaridae, I apologize, Cercaridae, Metasarcaridae, Cercaridae. They then hatch as Cercaridae, which are these kind of interesting, motile, free-swimming parasites that actually, where here the cyst is consumed, hatches in the gut. These actually free swim around, look for tadpoles, and then enter the tadpole at the location where the limb buds are forming. So they actually penetrate into the tadpole, burrow into the tadpole, and then there they produce a number of these cysts, the Metasarcariae. Let me move myself again here to over here. Here's a microscope image of the Cercaridae with 100 microns for scale. So they're actually quite large. So the head of this life stage is probably 200 to 300 microns. That's decent size. That's pretty impressive, and pretty big to be penetrating into the tadpole. I was pretty surprised about that they're able to burrow in that way. And that's the source of this deformation of generating multiple limbs, is when that limb bud is disturbed. It doesn't quite know what to do, so it produces a bunch of different limbs trying to make a good one effectively. And so the bud knows it's been compromised, and so it gets you can think of like a stem cell, and it's like shooting off a bunch of different limbs. It knows it needs to make a limb, but then the Cercaria is disrupting the limb that's been created, so it then starts to bifurcate and make all these crazy limbs here. So there's been this observation of Pacific tree frogs suggesting that the parasite can cause deformities. And they did this by using glass beads to mimic the effects of Ribaria, and that also produced deformities. So the first question was really, could a small, hard thing create deformities in a tadpole? So they did that by using an analog. So instead of trying to do an experiment where they were infecting tadpoles with known quantities of the flatworm, they're just putting in an analog, these little glass beads, to see if just the physical structure of the trematode is what's causing the deformity, or if it's something else. And so by putting in a small glass bead, they were able to replicate the same outcome. So by putting in a glass bead, then it causes a deformity. And then Johnson's done a ton of work on this. And so then further studies were looking at deformities of these Pacific tree frogs occurred in ponds that also had the aquatic snail, Heliosoma, as an intermediate host of the parasite. And then they found that all frogs with deformed limbs had Ribaria cysts. And so they're starting to piece it together of, okay, what's needed for Ribaria to persist in the environment, which is a full life cycle for Ribaria, which includes snails. And so now we're starting to make sense of, okay, well, the physical impact of Ribaria can cause multiple limbs, and we see frogs with multiple limbs or limb deformity in ponds that have the intermediate host, the snail, and all of the deformed frogs also have Ribaria cysts, the Metacercariae, which are like, I think it's a silica coating, but they have this like hard coating around them, and so they can survive for a very long time as well. And so we've moved from this observation. So we have one mechanism, which is the physical presence of an object can cause limb deformity. We have an observation of limb deformity only happening in places that have the parasitic flatworm, Ribaria, and in places that the Ribaria can complete its entire life cycle. So we've made this observation in nature, and now we want to test it in a controlled setting. So we've gone from a general observation in nature to, can we start to tease apart some of the mechanisms behind this, some of the reasons why. And so to do this, they did a controlled experiment using experimental groups compared to a controlled group, which lacked the factors being tested. And so in this case, tree frog eggs were exposed to Ribaria parasites in the lab with 4 treatments: a control, no parasites, 16, 32, and 48 parasites per tadpole or per egg. So you can imagine these as like little beakers or jars on the counter. So you've got one that just has frog eggs, no parasites, one that has a frog egg, 16 parasites, 32 and 48 parasites. And they watch to see what would happen. So here's a graph describing what happened. I'll move me again here and we'll walk through it. At first, this graph's kind of funny, like the way that they plotted it. It's a bit funny, but it makes sense once we walk through it. So if we start here with the control, 0 parasites per tadpole. So this is our jar that has a tadpole in it and no parasites. The red line is surviving frogs that had deformities. So looking here, 0% of the frogs had deformities. So in the absence of Ribaria, frogs do not have deformities. In the absence of Ribaria, survival was about 90%, 87% or so. And this is why control is important, because if we assumed that in the absence of a parasite, survivorship would be a 100%, that's false, right? There's a certain percentage of eggs that don't survive to tadpoles into metamorphosizing, and metamorphose into a frog. So there's a certain percentage of loss there. That's what the control helps us analyze, both perhaps due to lab conditions, to other things. Like you can imagine this experiment, if they messed up the lab conditions and half of the tadpoles died in the control group, and if you didn't have that control group and you looked at your parasite data and you said, oh, lots of them are dying, well, that could be due to your lab conditions. And so the control helps with that. Then we move up to 16 parasites per tadpole, or if you have one tadpole per jar, 16 parasites per jar. And the number of surviving frogs with deformity goes way up to about 70%. Overall survivorship drops as well to about 70%. So here with 16 parasites per tadpole, we have a massive increase in the number of tadpoles with deformities, and a decent decline in the number that survive. We go to 32 parasites per tadpole. Now we're down to about 40% survivorship and 95% that have deformities. Moving up to 48. Mortality stays about the same at about 40%, but the number of frogs with deformities goes up to nearly 100%. And they pick these concentrations based on environmental conditions, meaning they're based on what's commonly observed in the field. And so they didn't pick these numbers at random. Now, if we move on, so in a vacuum, meaning in a world where it's just clean water, happy frogs, and parasites, this makes sense, right? There's a gradation of the number of parasites and a gradation of both mortality and deformity. But we don't exist in a vacuum, as there's many other factors at work. And one of those could be a contributing factor of pesticides. And so this next study used a natural gradation of 6 ponds, all that had the parasite, all that had Ribria, but only 3 had pesticide contamination. And so here, wood frog tadpoles were placed in cages in each pond, 3 that had mesh sizes that allowed parasites to enter and 3 that didn't. So here we've got 2 different factors we're looking at. We're looking at the presence of parasites or not, dictated by the size of the cage. And so you can put tadpoles in or eggs in a small cage where the parasites are not able to enter. You can put them in a larger mesh-sized cage where the parasites are allowed to enter. So we've got effectively parasites and no parasites. Given that all the ponds do have parasites, we're able to exclude the parasites. So this is like a classic exclusion experiment. And then we're also using the natural gradation of 3 ponds having pesticides and 3 ponds not. So we'll look at the results of that study here. So I made a nice little matrix here. So we've got pesticides present and no pesticides. Mesh size 75 micron and 500 micron. So remember back to the graph I showed, or the life cycle of Ribera and the Sarkaria stage being relatively large. The scale bar there was 100 microns. So a 75 micron mesh would exclude the Sarkaria because their head's probably over maybe 200, 150 microns wide. So it's not going to be able to fit through this small mesh. And so for tadpoles living in the small mesh, there were no deformed frogs when there were no pesticides present, and there are no deformed frogs when there are pesticides present, which could be evidence to say that pesticides alone are not sufficient to cause this deformity. Because in the absence of the parasite, with the presence of pesticides, we do not have deformed frogs. Then we can move to the 500 micron mesh size with no pesticides present. So the 3 ponds with no pesticides, about 4% were deformed. So in the presence of parasite, lack of pesticide, deformity occurs at we could argue, a low rate, 4%. For those 4% of the frogs, it's probably relatively severe. And then when pesticides are present with the 500 micron mesh, Ribera can enter. It can parasitize the frogs, and with pesticides present, the percentage of deformity goes up to 29%. So this is showing kind of a mixed effect, meaning you have to have the parasite to have the deformity, but parasites are much more effective at creating deformity in the presence of pesticides. And this work was actually published in PNAS, which is a very high-ranked journal. And all these studies some were in Science. Like, these are all very big impact studies. And relatively basic, right? Like, we're talking about taking some frogs, throwing them in cages, dumping them in 6 ponds. Like, pretty simple stuff, but really profound impacts. So starting to get the mechanisms and understanding both multiple lines of evidence, but then also mixed effects on what's causing these deformities. So now we've made an observation, and we're hinting at a mechanism. So meaning we've made this observation that in ponds with pesticides and parasites, deformity rates are vastly increased. That's an observation, but what is the mechanism? Like what about pesticides is allowing this to happen? What's the physiological underlying factor that's resulting in this observation of pesticides plus parasites equals lots of deformity? So then this is kind of where lab studies come in. So we've made this natural observation, this really good natural experiment, but now we want to get at some of the more minutia mechanisms behind it so we can understand what about pesticides causes or results in the parasite being more effective. To do this, the authors came up with the hypothesis that pesticides decrease the ability of frogs to resist infection of parasites. Okay, so from the natural observation, more pesticide plus parasite equals massive rates of infection. So in another lab experiment, tadpoles were reared in the presence of pesticides, had fewer white blood cells, indicating suppressed immune system, and the higher rate of Ribera cyst formation. So here are some data from that study. So the top graph we'll walk through. So this is number of eosinophilus, which is white blood cells. Just think of this as white blood cells, based on cells per 5,000 red blood cells. So we're normalizing it by the number of red blood cells. So this is, just look at this graph as the number of white blood cells relative to red blood cells. So it's normalized by the number of red blood cells. And the bottom is number of cysts per tadpole, number of the Metasarcaria. And we've got 4 groups: control, solvent control, low pesticide, and high pesticide. So control would be like straight pond water, clean pond water. Solvent control would be the pesticides are in a solvent, and so they're adding that solvent as well, absent of the actual pesticide itself. And then low and high pesticides are just the gradation of the concentration of pesticides. So looking at the control, let's walk through the white blood cells first. Both the control and the solvent control had values in the 30 to 35 range. So 30 to 35 white blood cells per 5,000 red blood cells. And when we move into the 2 pesticide treatments, low and high, that number drops to 20 to like 17 white blood cells per 5,000 red blood cells. So a decent drop. I mean, almost half the number of white blood cells. So a good indication that there is a immune response related to pesticides, rather that pesticides can impact the ability for frogs to have a healthy immune system. and then in the number of cysts per tadpole. So this is looking at the actual infection rate, the number of cysts per tadpole. For the control and the solvent control, 15 or so, and that number jumps way up for the pesticides, both the low and the high pesticides, are over 65, 70 here, and 50-ish here with a larger error bar. So these are quite a lot different. So they're starting to draw this mechanism where it's not just pesticides, the presence of pesticides, it's what the pesticides do to the frog that enables increased parasitism in the tadpoles, meaning the pesticide is reducing the white blood cell count, it's reducing the ability for the frog to use its natural immune system to fight off these parasites. And so this is tied to the generation of the creation of synthetic pesticides, which began in the 1930s, and their use has increased dramatically. Obviously, we've gone through different versions of pesticides through time, and the amphibian exposure to pesticides has likely also increased, helping explain the increase in limb deformities, meaning that where we use more pesticide, it's fair to assume that frogs are also exposed to these pesticides. Fertilizer may also be a factor. So looking at the overall pond ecology, fertilizer runoff increases algal growth, which makes the snails that harbor Riberia happy because they eat algae. So more snails could result in greater numbers of Riberia. Now that's assuming that snail populations are the limiting factor to Riberia populations, which I don't know that we know. Meaning if you have a pond that has, one snail per meter squared, is that controlling the number of Riberia cysts that are in that pond? The number, in this case, Sarcariae, the little motile life stage. Or if we have 10 snails per meter squared, would we have more Sarcariae in that pond? I don't know that that's totally known, but mechanistically it kind of makes sense. But for that to be true, the snails have to be the limiting life stage. So it would make sense. With fertilizer runoff, there's a bunch of other factors, low DO, things like that could also negatively impact it. But it does make sense that there's multiple. The eutrophication also has a role to play, and it could also be stressing the tadpoles, which would result in more infestation. So when people alter one aspect of the environment, it can cause changes that we don't intend or anticipate. When we increase the use of pesticides and fertilizers, we don't then intend there to be deformity in frogs, but that's what we've done. Meaning there's unintended consequences that when we do an action, we don't fully understand. Often because it's not direct, right? Like, what does pesticides have to do with parasitic worms going after frogs? Like, it doesn't make any sense. But when we think, what are these pesticides doing in the environment? Well, one thing they could be doing is decreasing the immune system strength of frogs. Then it makes sense that they're more susceptible to lots of things, and one of those things happens to be parasites. So that's the first part of chapter 1. The second lecture will go through the last half of chapter 1. So thank you.